All right, kids, today we're going to get into our one-trait crosses. So before we move on, pause the video, take a look at our learning goals and success criteria for today. All right, so our joke of the day is, what did Mendel say when he had his breakthrough discovery in genetics? Whoopee! Get it? He used peas to do his experiments. All right, so one-trait crosses goes back to our meiosis. We got to think about meiosis when we have every cell that has two alleles for each gene. So there are two possible outcomes of meiosis. So for example, when you flip a coin, the probability of landing on heads is going to be 50%, and obviously it's going to be 50% for tails as well. So now we can combine that probability of landing heads twice, and that would decrease that chance to 25%. Now, we have something that's called a Punnett square, and that's what we're going to be using today. And it's developed by a British geneticist called Reginald Punnett. And what it does is it actually visually allows us to see what the traits look like when we cross them between two parents given the gametes. So what we're going to do is we're going to follow these steps to do a one-trait cross. First, you're going to draw a square or a rectangle, and you're going to divide it into 4 boxes. Okay, so it's good to do it with a large square so that you can fill in all the letters later on, which we're going to need to. Next, in the above squares, so on top of each column, you're going to put the gametes of one of the parents. So in our example right now, our gametes are a dominant R and a recessive R. So this is a heterozygous individual. And then on the other side, on the left side, we're going to put the other parent. So now we have the gametes of both parents. We can see that it is a monohybrid cross. Okay. So what happens is in the next steps, all you're going to do is you're going to bring these letters together. Okay. So these letters will come down here. So if it's on top of a column, it's just going to drop to each of the boxes below it. So this R will drop down to here. This R drops down to here. The big R drops down to here. The big R drops down to here. Okay. And then what you're going to do is on this side, you're going to move all the letters to every box into the row to the right of it. So this big R is going to be here. Big R, little r, little r. So what it's going to look like is like that. Our result is, after we put all the letters together, see how here it's exactly what we did. It corresponded to what's on top of each column and on the side of the row, and we're going to have our results here. And based off of what we see in our results here, it'll tell us exactly what our genotypes are. And based on the genotypes, we'll be able to tell what our phenotypes are going to be. So that shows exactly how Mendel's law of segregation happens exactly, right? Because we have each gamete will segregate separately and has equal chances to go into the offspring that is in the next generation. So visibly we can see it, and because there's 4 squares, that means each square is 25% chance of showing up in the next generation. So let's take a look. Another example here, this one's done for us. It is exactly the same as what we kind of looked at, 2 heterozygous parents coming together. Now let's look at our chances or our probability here. We have a purple flower that we're looking at. I know it looks kind of blue, but this P for purple. Okay, so we have 1 homozygous dominant. I know it doesn't really fit in here. Sorry about that. We have 2 heterozygous. And then we have 1 recessive. Okay, so this will show up because we remember from the last lesson that if it is heterozygous and homozygous, it'll show up as the more dominant trait. And then for the recessive trait to show up, it's going to have to be a homozygous recessive. So this Punnett square shows a monohybrid cross, okay, one trait cross between two heterozygous F1 generation purple flowers. When we say monohybrid, that means there's only one trait that we're looking at, but they're hybrid. So that means they have to be heterozygous if they're hybrid. So the result is going to be a prediction of genotypes and phenotypes of the F2 generation. And what we saw, okay, is our F2 generation would show up as 3 purple and 1 white. And for genotypes, it is going to be 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. All right, so what are test crosses? Test crosses are between an individual of unknown genotype for a trait and an individual that is homozygous recessive for that trait. So we know that homozygous recessive is going to show up only if there's 2 alleles for the recessive trait. Now, if it is showing up as the dominant trait, we don't know whether or not it's heterozygous or homozygous dominant. So what we're going to need to know is how to use a test cross to figure that out. So by analyzing the phenotype of the offsprings, we will be able to provide information of the unknown genotype of the parent. So here's our example. A breeder could do a test cross between purple flower pea and a white flower pea plant. Now we don't know what color or what genotype it is. It shows up as purple, but it could be heterozygous or homozygous dominant. So a test cross can be done. So in the next slide right here, we have a test cross between, so here's our homozygous recessive here, and our other parent, who the 2 scenarios that are given are, one is the homozygous dominant, 2 uppercase, right here, and then the 2 lowercase, or the heterozygous over here. Now, because you cross it, let's look at our results. All 4 of them are going to show up as purple, right? They're all heterozygous. So if it's a homozygous dominant parent with a homozygous recessive parent, we're going to get 100% purple. And if it is a heterozygous parent, you're going to get 2 purple and 2 whites. So when we look at the ratios, we can actually reduce that to 1 to 1. So that goes back to our question. Even if one of the offspring are white, why can you not be absolutely sure that the genotype of the unknown parent is homozygous dominant? Well, we just talked about it, is because it will show up as dominant color doesn't necessarily mean is homozygous dominant. It could also be heterozygous because the hidden allele can be there, it's just not expressed at the time. All right, so let's take a look at our sample problem. Sample problem A. In pea plants, the allele for round seeds is dominant to allele for wrinkled seeds. If a pea plant that is heterozygous for seed shape is crossed with a pea plant that is all right, so sample problem A, in pea plants, the allele for round seeds is dominant and the allele for wrinkled seeds is recessive. Okay, so if it says dominant to something, so that means the first one is dominant and the second one is going to be recessive. So if a pea plant that is heterozygous for seed shape crosses with a pea plant that produces wrinkle seeds, determine the following. So we got to find out what the genotypes of the P generation plants are, and then we got to find out what the F1 generation plants are going to be. So how we're going to do this is like this. First thing we want to do is I like to highlight or write down exactly what we're looking at. So we know that round seeds is dominant and then wrinkle seeds is going to be recessive. So let's write that down. I'm going to write down your dominant. So because it's round, it's dominant, we're going to use the first letter of the dominant allele. So R. is going to be round. So uppercase R round, lowercase for the recessive means wrinkled. Now with that, let's take a look at more clues that they've given us. If a plant is heterozygous for seed shape, that's crossed with wrinkle seeds. So heterozygous, we know that right away it's going to be one dominant and one recessive allele. So it's the first parent is going to be like that. And for something to express as wrinkled, and we know that wrinkled is recessive, so that means it's going to have to cross with our homozygous recessive, because the only way it'll show up if the recessive allele is going to show up is if it's homozygous recessive. So here's what we're going to be crossing. Now, we're ready to cross. By the way, we've answered our first question, the genotypes of the P generation plants. Right here. Here are genotypes. Now, from that, we actually can figure out what the gametes are from the last, the pre-lesson 6 that I posted on the Google Classroom. So we're just going to move on from there. Let's cross these together. So we're going to put our gametes up here. Actually, you know what? Let me use a different color here, just so that we can differentiate what we're looking at. So let's use a nice blue color. So our gametes for our first parents on the top column, you could put it on the side, doesn't matter. If you reverse them, it's okay. And then we're going to put the gametes of the other parent. Now we're going to fill our boxes. When we put our genotypes, we're going to put all the same. So if they're capital, I like to put them first. And then if they are lowercase, then I'll put them second. Okay, so in this case, I'm going to put all my capital letters. So all my uppercase goes first. So I don't want to put the lowercase first and then the uppercase, okay? That way we'll be able to look at it a little bit more in an organized way, and it's easier to tell what we're looking at. All right, so here's our results, and I've actually done an answer page, next page. So our results are here. They're exactly the same compared to the one before. I put the heterozygous parent on the top. Whether or not you put it on the side here or on the top, it doesn't matter. The results are going to be the same. We have 2 heterozygous round and 2 homozygous recessive. So that means we have a 50-50 chance at a round and 50-50 chance of a wrinkled. So here it's asking us, let's go back to the question, the genotypes and phenotypes. Okay, so genotypes, we already got it here. And the phenotypes is going to be 2 round, 2 wrinkled. And then it's asking us what happens to the next batch of offspring. Well, if it's the same parent, the offspring probability is still going to be the same. It's still going to be 50-50. So it's going to have a 50% chance of having a wrinkled seed offspring. All right, second sample problem. This one requires a little bit of logic, but let's take a look at this. In pigs, the allele for black coat color is dominant to the allele for white coat color. 2 pigs mate, producing 5 black piglets and 1 white piglet. We're going to determine the genotypes of parents and the probability of producing a white piglet in the next litter. So here's an example of a question that doesn't necessarily need to start off with a Punnett square. We got to figure out what the alleles are for both parents first. So let's take a look. I'm going to bust out my highlighter. So allele for black coat is dominant to the allele for white coat. So that means if our dominant allele is black, first letter, we're going to take our first letter is B. So uppercase B represents black. And lowercase b represents white. Okay, so our clues are 2 black pigs mate. And there's 5 black piglets and one white piglet that's being made here. Now we got to figure out what the genotypes of the 2 parents. So the 2 parents are 2 black pigs. Now we don't know what the genotypes are, but we know from the clues that are here given to us what their offspring look like. Now, for something to show up as recessive, because we know that white is recessive, for it to show up as recessive, it's got to be homozygous recessive, right? And it gets those homozygous recessive alleles, one from each parent. So that means both parents have to have a recessive allele in there that's not expressed. So now we have it. The answer is right here for number 1, determine genotypes. We know that it shows up as black. So let's cross 2 here. Now we don't know what that second one is. As I was saying before, our white piglet will get the alleles from both parents, so that means their recessive allele has to be from each parent. So therefore each parent has to be heterozygous. And here it is, genotype for both parents are heterozygous. Now, determine the probability of producing a white piglet in the next litter. Next litter means it's the same parents. So now we have to take out our Punnett squares again. Going to switch color for our ink. All right, let's put pen, ink color. Okay, let's put our gametes here. Now we're going to have the entire thing filled out. So here are your results. I actually have an answer slide again for this. Here are results. We have 2 black pigs. They're going to produce white piglets. So that means they both have recessive alleles. That means they both have to be heterozygous. Okay. And it's asking us the next litter. What are the chances that's going to be white? Well, this is going to be black. This is black. This is black. This is the only white. So 1 in 4, right? Because there's 1 white, 3 black. So that is 1 in 4 going to be white litter. So that means 25%. All right. So that's Punnett squares, the intro to Punnett squares in a nutshell. I have posted some of the questions on the Google Classroom in the same task that I'm giving you today. Please try to do these questions and make sure you ask me if you have any questions on this stuff. Okay, so find me either in the office hours, email, or Q&A. Remember that email, Q&A, or if you post anything that's on Google Classroom, it might be delayed. So if you want an instant answer, make sure you go on to the office hours, because I will be on every day at 9 o'clock to 10 o'clock sharp. All right, so try the questions out. If you have any questions, let me know. And that is all for today, folks.